Skeletal muscle fibers. Each skeletal muscle fiber is a single, long, cylindrical cell containing several nuclei, which are located around its periphery near the plasma membrane. A single fiber can extend from one end of the muscle to the other. In most cases, the fibers range from approximately 1 millimeter to about 4 centimeters in length and from 1 micrometer to 100 micrometers in diameter. Large muscles contain a large percentage of large diameter fibers, whereas small, delicate muscles contain a large percentage of small diameter fibers. However, any given muscle contains a mixture of small and large diameter fibers, as seen in the longitudinal section. Alternating light and dark bands give the muscle fiber a striated or striped appearance. Figure 9.2. Origin of muscle fibers. Muscle fibers develop from less mature multinucleated cells called myoblasts. The multiple nuclei result from the fusion of myoblast precursor cells, not from the division of nuclei within myoblasts. Myoblasts are converted to muscle fibers to contractile proteins accumulate within their cytoplasm. Shortly after the myoblasts form, nerves grow into the area and innervate the developing muscle fibers. The number of skeletal muscle fibers remain relatively constant after birth. Enlargement or hypertrophy of muscles after birth in children and adults result from an increase in the size of each muscle fiber, not from the substantial increase in the number of muscle fibers. Similarly, hypertrophy of muscle in response to exercise is due mainly to an increase in muscle fiber size rather than the increase in number. Histology of muscle fibers. Muscle contraction is much easier to understand when we consider the structure of a muscle fiber. 9.3. The plasma membrane of a muscle fiber is called the sarcolemma. Two delicate connective tissue layers are located just outside the sarcolemma. The deeper and thinner of the two is the external lamina. It consists mostly of reticular collagen fibers and is so thin that it cannot be distinguished from the sarcolemma when viewed under a light microscope. The second layer also consists mostly of reticular fibers, but it is a much thicker layer called the endomysium. Along the surface of the sarcolemma are many tube-like innervations of the sarcolemma called transverse tubules or T-tubules. They occur at regular intervals along the muscle fiber and extend inward, connecting the extracellular environment with the interior of the muscle fiber. 9.3b. The T-tubules are also associated with the highly organized, smooth endoplasmic reticulum called the sarcoplasmic reticulum. In skeletal muscle fiber, other organelles such as numerous mitochondria, energy-storing glycogen granules, are packed into the cell and constitute the cytoplasm, which in the muscle cell is called the sarcoplasm. The sarcoplasm also contains numerous myofibrils, which are bundled of protein filaments. Each myofibril is a thread-like structure, approximately 1 to 3 micrometers in diameter, that extends from one end of the muscle fiber to the other. A myofibril contains two kinds of protein filaments called myofilaments. 9.3c. Actin myofilament or thin myofilaments are approximately 8 nanometers in diameter and 1,000 nanometers in length, whereas myosin myofilaments are thick myofilaments are approximately 12 nanometers in diameter and 1,800 nanometers in length. The actin and myosin filaments form highly ordered units called sarcomeres, which are joined end to end to form myofibrils. 9.4a. Actin and myosin myofilaments. Each actin myofilament is composed of two strands of fibrous actin, F-actin, a series of tropomyosin molecules, and a series of troponin molecules. The two strands of F-actin are coiled to form a double helix, which extends the length of the actin myofilament. Each F-actin strand is a polymer of approximately 200 small globular lumens called globular actin G actin monomers. Each G-actin monomer has an active site to which myosin molecules can bind during muscle contraction. Tropomyosin is an elongated protein that winds along the groove of the F-actin double helix. Each tropomyosin molecule is sufficiently long to cover 7 G-actin active sites. Troponin is composed of 3 subunits, one that binds to actin, a second that binds to tropomyosin, and a third that binding site for calcium. The troponin molecules are spaced between the ends of the tropomyosin molecule and the groove between the F-actin strands. The complex of tropomyosin and troponin regulate the interaction between active sites on G-actin and myosin. Myosin filaments are composed of many elongated myosin molecules shaped like golf club heads. 9.4b and c. Each myosin molecule consists of 2 myosin heavy chains wound together to form a rod portion lying parallel to the myosin filament and 2 myosin heads that extend laterally. 9.4b, see figure 9.3d. 4 light myosin chains are attached to the heads. Excuse me. Excuse me again. Attached to the heads of each myosin molecule. Each myosin myofilament consists of about 300 myosin molecules arranged so that about 150 of them have their heads projected towards each end. The centers of the myosin myofilaments consist of only the rod portions of the myosin molecule. The myosin heads have 3 important properties. 1. The heads can bind to active sites on the actin molecules to form cross bridges. 2. The heads are attached to the rod portion by a hinge region that can bend and straighten during contraction. And 3. The heads are ATPase enzymes which break down adenosine triphosphate ATP releasing energy. Part of the energy is used to bend the hinge region of the myosin molecule during contraction. Sarcomeres. The sarcomere is the basic structural and functional unit of skeletal muscle. Because of the smallest portion of skeletal muscle capable of contracting, each sarcomere extends from one Z-disc to an adjacent Z-disc, figures 9.4 and 5. A Z-disc is a filamentous network of protein forming a disc-like structure for the attachment of actin myofilaments. The arrangement of the actin myofilaments and the myosin myofilaments give the myofibril a banded or striated appearance when viewed longitudinally. Each isotropic band or I-band includes a Z-disc and extends from each side of the Z-disc to the ends of the myosin myofilaments. When seen in longitudinal and cross sections, the I-band on each side of the Z-disc consists only of actin myofilaments. Each anisotropic band or A-band extends the length of the myosin myofilaments within a sarcomere. The actin and myosin myofilaments overlap for some distances at both the ends of the A-band. In a cross-section of the A-band where actin and myosin myofilaments overlap, each myosin myofilament is visibly surrounded by 6 actin myofilaments. In the center of each A-band is a smaller band called the H-zone, where the actin and myosin myofilaments do not overlap and only myosin myofilaments are present. A dark line called the M line is in the middle of the H-zone and consists of a delicate filament that attaches to the center of the myosin myofilaments. The M line helps hold the myosin myofilaments in place, similar to the way the Z-disc holds actin myofilaments in place. Figure 9.5, B and C. The numerous myofibrils are orientated within each muscular fiber so that A-bands and I-bands are parallel myofibrils are aligned and thus produce a striated pattern when seen through a microscope. In addition to actin and myosin, there are other less visible proteins within sarcomeres. These proteins help hold actin and myosin in place, and one of them accounts for muscles' ability to stretch extensibility and to recoil elasticity. Titin is one of the largest known proteins, consisting of a single chain of nearly 2,700 amino acids. It attaches to the Z-disc and extends along myosin myofilaments to the M line. The myosin myofilaments are attached to the titin molecules, which help hold them in position. Part of the titin molecule is the I-band functions as a spring, allowing the sarcomere to stretch and recoil. The primary function of skeletal muscle cells is to generate force by contracting or shortening. A parallel arrangement of myofilaments and sarcomere allows them to interact, which causes muscle contraction. The interaction is described by the sliding filament model. figure 9.6. Sliding filament model. It is the shortening of sarcomeres that is responsible for the contraction of skeletal muscle. In a sarcomere, the actin and myosin myofilaments slide past one another but remain the same length as when the muscle is at rest. When the myofilaments slide past each other and the sarcomeres shorten, the myofibrils also shorten because the myofibrils consist of sarcomeres joined end to end. The myofibrils extend the length of the muscle fibers, and when they shorten, the muscle fibers shorten. Groups of muscle fibers make up a muscle vesicle, and several muscle vesicles make up a whole muscle. Therefore, when sarcomeres shorten, myofibril muscle fibers, muscle vesicles, and muscles shorten produce muscle contraction. During muscle relaxation, the sarcomeres lengthen. For this to happen, an external force must be applied to a muscle by other muscles or by gravity. For example, muscle contraction causes the joint, such as the elbow or knee, to flex. Extension of the joint and lengthening of the muscle result from the contraction of muscles that produce the opposite movement. See Chapter 10. However, knowing the mechanism by which muscles shorten is a small step towards understanding muscle contraction. What stimulates the sarcomeres to shorten in the first place? In order to fully understand muscle contraction, we must first consider the electrical properties of skeletal muscle fibers. Muscle fibers, like other cells in the body, are electrically excitable. A description of the electrical properties of skeletal muscle fibers is presented next, and later sections illustrate the role of these properties in contraction. Axons and neurons extend from the brain and spinal cord to skeletal muscle fibers. The nervous system controls the contraction of skeletal muscle through these axons. Electrical signals called action potentials travel from the brain to spinal cord along the axons to muscle fibers and cause them to contract. Ion channels. In order to understand electrical properties of skeletal muscle fibers, a review of the permeability characteristics of cell membranes and the role membrane transport proteins play in the permeability will be helpful. The phospholipid bilayer interior is a hydrophobic environment which inhibits the movement of charged particles, particularly ions, across the membrane. However, the basis of the electrical properties of skeletal muscle cells is the movement of ions across the membrane. Recall from Chapter 3 that ions can move across a membrane through ion channels. There are two major types of ion channels, non-gated or leaked and gated. These two channel types contribute to the electrical properties of both resting cell and stimulated cell. In resting cells, the non-gated ion channels allow for the slow leak of ions down their concentration gradient. Like all membrane transport proteins, leak channels are specific for a particular ion. The gated ion channels are more important in stimulated cells. It is their presence that governs the production of action potentials. There are two major gated ion channels. Ligand-gated ion channels. Ligand-gated ion channels open when a ligand or chemical signal binds to a receptor that is part of the ion channel. See Figure 3.8. For example, the axons of neurons supplying skeletal muscle fibers release ligands called neurotransmitters, which bind to ligand-gated Na+ channels in the membranes of the muscle fibers. As a result, the Na+ channels open, allowing Na to enter the cell. Voltage-gated ion channels. These channels are gated membrane channels that open and close in response to a particular membrane potential. When a neuron or muscle fiber is stimulated, the charge difference changes, and particular change causes certain voltage-gated ion channels to open or close. The voltage-gated ion channels that play major roles in the action potential are voltage-gated Na+, K+, and Ca2+ channels. For example, opening voltage-gated Na+ channels allow Na to cross the plasma membrane, whereas opening voltage-gated K channels allow K+ to cross potassium, and opening Ca2+ calcium channels allow Ca2+ to cross. Differences exist across each plasma membrane. Resting membrane potential. Electrically excitable cells, like most cells, are polarized. That is, the inside of the most plasma membranes is negatively charged compared to the outside. Thus, a voltage difference or electrical charge difference exists across each plasma membrane. This charge difference across the plasma membrane of an unstimulated cell is called a resting membrane potential. Figure 9.7, see Figure 11.14. Action potentials cannot be produced without a resting membrane potential. The resting membrane potential is the result of three factors. 1. The concentration of K plus potassium inside the plasma membrane is higher than that outside the plasma membrane. 2. The concentration of Na plus outside the plasma membrane is higher than inside the plasma membrane. And 3. Plasma membrane is more permeable to K plus than to Na plus because the concentration gradient for the ion determines whether the ion enters and leaves the cell after its ion channel opens. When voltage-gated Na plus channels open, Na plus moves through the channels into the cell. In a similar fashion, when gated K plus channels open, K plus moves out of the cell. Since excitability cells have many K plus leak ion channels, K plus moves out of the cell faster than Na plus moves into the cell. In addition, negatively charged molecules such as proteins are trapped inside the cell because plasma membranes are impermeable to them. For these reasons, the inside of the plasma membrane is more negatively charged than outside. Some K plus is able to diffuse down the concentration gradient from inside to just outside the plasma membrane because K plus is positively charged, and its movement from inside the cell to outside causes the inside of the plasma membrane to become even more negatively charged compared with outside. Potassium ions diffuse down the concentration gradient only until the charge difference across the plasma membrane is great enough to prevent any additional diffusion of K plus out of the cell. The resting membrane potential is in equilibrium with the tendency for K plus to diffuse out of the cell is opposed by the negatively charged inside of the cell, which tend to attract the positive K plus into the cell. At rest, the sodium-potassium pump transports K plus from outside the cell to inside and it transports Na plus from inside the cell to the outside. The act of transports Na plus and K plus by the sodium-potassium pump maintains the uneven distribution of Na plus and K plus across the plasma membrane. See Chapter 3. The details of the resting membrane potential are described more fully in Chapter 11. The resting membrane potential can be measured in units called millivolts. The potential difference across the plasma membranes of neurons and muscle fibers are between negative 70 and negative 90 millivolts. The potential difference is reported as a negative number because the inner surface of the plasma membrane is negative compared with the outside. Action potential. An action potential is a reversal of the resting membrane potential such that the inside of the plasma membrane becomes positively charged compared with the outside. The permeability characteristics plasma membrane change because ion channels open when a cell is stimulated. The diffusion of ions through these channels changes the charge across the plasma membrane and produces action potential. An action potential lasts for approximately 1 millisecond to a few milliseconds, and it has two phases, depolarization and repolarization. Figure 9.8 depicts the changes that occur in the membrane potential during an action potential. Stimulation of the cell can cause its plasma membrane to become depolarized. Specifically, the inside of the plasma membrane becomes less negative as indicated by movement of the curve upward towards zero in figure 9.8a. If the depolarization changes the membrane potential to a value called threshold, an action potential is triggered. The depolarization phase of the action potential is a brief period during which further depolarization occurs and the inside of the cell becomes positively charged. 9.8b. The charge difference across the plasma membranes is said to be reversed when the membrane potential becomes a positive value. Repolarization phase is the return of the membrane potential to its resting value. The opening and closing of voltage-gated ion channels change its permeability of the plasma membrane to ions, resulting in depolarization and repolarization. Before a neuron or a muscle fiber is stimulated, these voltage-gated ion channels are closed. 9.9. Step 1. When the cell is stimulated, gated Na plus channels open and Na plus diffuses into the cell. The positively charged Na plus makes the inside of the plasma membrane less negative. If this depolarization reaches threshold, many high voltage Na plus channels are stimulated and open rapidly, causing Na plus to diffuse into the cell until the inside of the membrane becomes positive for a brief time. 9.9. Step 2. As the inside of the cell becomes positive, this voltage changes causes additional permeability changes in the plasma membrane, which stop depolarization and start repolarization. Repolarization results from the closing of the voltage-gated Na plus channels and the opening of the voltage-gated K plus channels. 9.9. Step 3. Thus, Na plus stops moving into the cell and more K plus moves out of the cell. These changes cause the inside of the plasma membrane to become more negative and the outside to become more positive. The action potential ends and the resting membrane potential is reestablished when the voltage-gated K+ channels close. Action potentials occur according to the all or none principle. If a stimulus is strong enough to produce depolarization that reaches threshold, or even if it extends threshold by a substantial amount, all of the permeability changes are responsible for the action potential proceed without stopping. Consequently, all of the action potentials in a given cell are alike, the all part. If a stimulus is so weak that the depolarization does not reach threshold, few of the permeability changes occur. The membrane potential returns to its resting level, and after a brief period without producing an action potential, the none part. An action potential can be compared to the flash system of a camera. Once the shutter is triggered, reaches threshold, the camera flashes. An action potential is produced, and each flash is the same brightness, the all part, as the previous flash. If the shutter is depressed but is not triggered, does not reach threshold, the flash results, no flash results, the none part. An action potential occurs in a very small area of the plasma membrane and does not affect the entire plasma membrane at one time. Action potentials can propagate across the plasma membrane because an action potential produces at one location in the plasma membrane can stimulate the production of an action potential in an adjacent location, figure 9.10. Note that an action potential does not actually move along the plasma membrane. Rather, an action potential at one location stimulates the production of another action potential in an adjacent location, which in turn stimulates the production of another, and so on. It is much like how a row of toppling dominoes in which each domino knocks down the next. Each domino falls, but no single domino actually travels the length of the row. The action potential frequency is the number of action potentials produced per unit of time. As the strength of the stimulus applied to a neuron or a muscle fiber increases once threshold is reached, the action potential frequency increases as the strength of the stimulus increases. All of the action potentials are identical. The action potential frequency can affect the strength of a muscle contraction. See stimulus frequency and whole muscle contraction in section 9.5. In summary, the resting membrane potential results from a charge difference across the plasma membrane. An action potential, which is reversal of that charge difference, stimulates cells to respond by contracting. The nervous system controls muscle contractions by sending action potentials along axons to muscle fibers, which then stimulate action potentials in the muscle fibers. An increased frequency of action potentials sent to the muscle fibers can result in a stronger muscle contraction. Neuromuscular junction. Action potentials carried by motor neurons stimulate action potentials in muscle fibers because of events that occur in neuromuscular junction. Axons of motor neurons carry action potentials at a high velocity from the brain and spinal cord to the skeletal muscle fibers. The axons branch repeatedly, and each branch projects towards one muscle fiber to innervate it. Thus, each muscle fiber receives a branch of an axon, and each axon innervates more than one muscle fiber. See figure 9.1. Near the muscle fiber it innervates, each axon branch forms a cluster of enlarged axon terminals that result in an invaginization of sarcolemma to form a neuromuscular junction or synapse. Therefore, a neuromuscular junction consists of the axon terminals and the area of the muscle fiber sarcolemma they innervate. Each axon terminal is the presynaptic terminal. The space between the presynaptic terminal and the muscle fiber is the synaptic cleft, and the muscle plasma membrane in that area of the junction is the postsynaptic membrane or motor end plate, figure 9.11. Each presynaptic terminal contains numerous mitochondria and many small spherical sacs approximately 45 micrometers in diameter called synaptic vesicles. The vesicles contain acetylcholine, 45 nanometers, acetylcholine, ACH, an organic molecule composed of acetic acid and choline. Acetylcholine is a neurotransmitter, a substance released from a presynaptic membrane that diffuses across the synaptic cleft and alters the activity of the postsynaptic cell. Neurotransmitters can stimulate or inhibit the production of an action potential in the postsynaptic membrane or sarcolemma by binding to ligand-gated ion channels. When an action potential reaches the presynaptic terminal, it causes voltage-gated calcium ion Ca2+ channels in the plasma membrane of the axon to open. As a result, Ca2+ diffuses into the cell, figure 9.12. Once inside the cell, the Ca2+ causes the contents of a few synaptic vesicles to be secreted by exocytosis from the presynaptic terminal into the synaptic cleft. The acetylcholine molecule releases from the synaptic vesicles, then diffuse across the cleft and bind to the receptor molecules within the postsynaptic membrane, thus causing ligand-gated Na+ channels to open, increasing the permeability of the membrane to Na+. Sodium ions then diffuse into the cell, causing depolarization in skeletal muscle. Each action potential in the motor neuron causes depolarization that exceeds threshold, which causes changes in voltage-gated ion channels that produce an action potential in the muscle fiber. Acetylcholine release into the synaptic cleft is rapidly broken down to acetic acid and choline by the enzymes acetylcholine esterase, figure 9.12. Acetylcholine esterase keeps acetylcholine from accumulating within the synaptic cleft, where it would act as a constant stimulus to the postsynaptic terminal, producing many action potentials and continuous contraction in the muscle fiber. The release of acetylcholine and its rapid degradation in the synaptic cleft ensures that one presynaptic action potential yields only one postsynaptic action potential. Choline molecules are actively reabsorbed by the presynaptic terminal, then combined with acetic acid produced in the cell to form acetylcholine. Recycling acetylcholine molecules requires less energy and is more rapid than repeatedly synthesizing new acetylcholine molecules each time they are released from their presynaptic terminal. Acetic acid is an intermediate in the process of glucose metabolism. See chapter 25. A variety of cells can take it up and use it after it diffuses from the area of the neuromuscular junction. Excitation, contraction, coupling. Action potentials produced in the sarcolemma of a skeletal muscle fiber can lead to the contraction of the fiber. The mechanism by which an action potential causes contraction of a muscle fiber is called excitation, contraction, coupling, and it involves the sarcolemma. T-tubules and sarcoplasmic reticulum Ca2+ and troponin T-tubules project into the muscle fiber and wrap around sarcomeres in the region where the actin and myosin myofilaments overlap, figure 9.13, see figure 9.3. The lumen of each T-tubule is filled with extracellular fluid and is continuous with the exterior of the muscle fiber. Near the T-tubules, the sarcoplasmic reticulum is enlarged to form terminal cisternae. A T-tubule and the two adjacent terminal cisternae together are called a triad. Thus, sarcoplasmic reticulum actively transports Ca2+ into the lumen. Thus, the concentration of Ca2+ is approximately 2,000 times higher within the sarcoplasmic reticulum than the sarcoplasm of the resting muscle fiber. Excitation contraction coupling begins at the neuromuscular junction with the production of an action potential in the sarcolemma. The action potential is propagated along the entire sarcolemma of the muscle fiber and into the T-tubules. The T-tubules carry action potentials into the interior of the muscle fiber, where they cause voltage-gated Ca2+ channels in the terminal cistern of the sarcoplasmic reticulum to open. When Ca2+ channels are open, Ca2+ rapidly diffuses into the sarcoplasm surrounding the myofibrils, figure 9.14. Calcium ions bind to the Ca2+ binding sites on the troponin molecules of the actin myofilaments. The combination of Ca2+ with troponin causes the troponin-tropomyosin complex to move deeper into the groove between the two F-actin strands, which exposes active sites on the actin myofilaments. The head of the myosin molecules then bind to the exposed active sites to form cross bridges, figure 9.14. Movement of the cross bridges results in contraction. Cross bridge movement. A cycle of events resulting in contraction proceeds very rapidly. Figure 9.15. The heads of myosin molecules move at their hinge region, resulting in the cross-bridge movement. This movement forces the actin myofilaments to which the heads of the myosin molecules are attached to slide over the surface of the myosin myofilament. After cross-bridge movement, each myosin head releases from the actin and returns to its original position. It can form another cross-bridge at a different site on the actin myofilament, following by movement release of the cross bridge and return to its original position. During a single contraction, each myosin molecule undergoes a cycle of cross-bridge formation, movement, release, and return to its original position many times. This process is called cross-bridge cycling. The energy from one ATP molecule is required for each cross-bridge formation, the movement and release. Before a myosin head binds to the active site on an actin myofilament, the head of the myosin molecule is in its resting position, and ADP and phosphate are bound to the head of the myosin molecule, figure 9.15. Once Ca2 plus binds to the troponin and the tropomyosin moves, the active sites on actin myofilaments are exposed. The head of the myosin molecule can then bind to an exposed active site, and phosphate is released from the head of the myosin molecule. Energy stored in the head of the myosin molecule causes the head of the myosin molecule to move. Movement of the head causes the actin myofilament to slide past the myosin myofilament, and ADP is released from the myosin head. ATP must then bind to the head of the myosin before the cross bridge can release. As the ATP molecule binds to the head of the myosin head breaks ATP apart into ADP and phosphate, and the myosin head releases from the active site on the active. The ADP and phosphate remain attached to the head of the myosin molecule, and the head of the myosin returns to its resting position. The energy released from the breakdown of ATP is stored in the head of the myosin molecule. Movement of the myosin molecule while the cross bridge attached is called the power stroke, whereas return of the myosin head to its original position after cross bridge release is called the recovery stroke. Many cycles of power and recovery strokes occur during each muscle contraction. While muscle is relaxed, energy stored in the heads of the myosin molecules held in reserve until the next contraction. When Ca2 plus is released from the sarcoplasmic reticulum in response to an action potential, the cycle of cross-bridge formation, movement, and release, which results in contraction, begins. See figure 9.14 and 15. Figure 9.16 summarizes the overall sequence of events that occurs as a stimulus causes skeletal muscle to contract. Muscle relaxation. Muscle relaxation occurs when acetylcholine is no longer released at the neuromuscular junction. The cessation of action potentials along the sarcoplasmic stops. Ca2 plus release from the sarcoplasmic reticulum, and Ca2 plus is actively transported back into the sarcoplasmic reticulum. As the Ca2 plus concentration decreases in the sarcoplasm, the Ca2 plus diffuses away from the troponin molecules. The tropomyosin complex then reestablishes its position, which blocks the active sites on the actin molecules. As a consequence, the cross bridge cannot reform once they have been released, and the muscle relaxes. Thus, energy is needed not only to make muscle fibers contract, but also to make the muscle fibers relax. 3 major ADP-dependent events are required for muscle relaxation. After an action potential has occurred in the muscle fiber, the sodium-potassium pump must actively transport Na plus and K plus to return to and maintain resting membrane potential. ADP is required to detach the myosin heads from the actin and return them to their resting position. The ATP is needed for the active transport of Ca2 plus into the sarcoplasmic reticulum. Because the reuptake of Ca2 plus into the sarcoplasmic reticulum is much slower than the diffusion of Ca2 plus out of the sarcoplasmic reticulum, a muscle fiber takes at least twice as long to relax as it does to contract.